So my name is Tina Bagratuni. I work at the University of Athens, and I have been selected for an IWMF grant, for which I'm presenting a couple of studies here at Milan at the EHA conference. So this is actually a study which is trying to understand the ibrutinib-resistant mechanism in patients that are undergoing ibrutinib therapy. So we know that about a third of patients are not responding to ibrutinib, and we wanted to check in a very, very sensitive way, in a very specific way, why this actually happens, why these patients are not responding to ibrutinib. So the way to approach this, we used a very much said sorry. We are trying to do a very high throughput technology, which is the single cell RNA sequencing analysis in combination with whole genome sequencing analysis. So for the whole genome sequencing analysis, we're trying to find which are the mutations that might be linked to the disease and the actual resistant mechanism. And in the same way, we're trying to do single cell RNA sequencing analysis, again, to try and identify the genes that are found in the non-responder patients and are not found in the responders, and vice versa. So in this way, we're going to basically focus on the B cell clone, as well as the tumor microenvironment. So one of the posters that are being presented this afternoon is actually focusing on all the single cell analysis that has been conducted and all of the whole genome sequencing analysis. So here we have actually done all of the analysis in about 37 patients, and about 14 patients are resistant to this to ibrutinib. And what we find actually is that the B cells are much higher in these non-responding patients. This means that actually the tumor cells are much higher, and these are linked to specific pathways, which are basically the NF-kappa B pathway, the ERK pathway, the Wnt pathway, and these are something that has been shown in the literature as well. We also find that there are specific mutations that are linked to this resistant mechanism. So we find that actually patients that are not responding to ibrutinib have mutations 6CR4 as expected. We do find many mutations in the KMT2D gene. We do also find mutations in the ARID1A gene. These are actually not found in the responding patients. So maybe this something drives this resistant mechanism only with the mutation signature as well. So what we're trying to do at the moment, this is not will not be seen in this conference. We're trying to combine now this whole genome sequencing analysis with the single cell analysis and see what mutations are linked to what gene expression, and come up with a signature which will help patients to understand what are these features that we can find at their diagnosis that will help them not have an ibrutinib treatment and maybe use an alternative treatment which will be more effective to these patients. The other poster that we are presenting here as well is the liquid biopsy. So here we're trying to sort the clonal B cells from these patients. Again, these are the same patients that were used in the first study. And what we're trying to see here is, by sorting them from the peripheral blood and sorting them from the bone marrow, if the mutation patterns that are found in the bone marrow reflect the ones that are found in the peripheral blood. So hopefully, if we find these similarities, then we can avoid the repetitive bone marrow tests that some patients need to undergo in terms to screening processes. So we find that there's a very good reflection. So there are many similarities among the sorted bone marrow, the sorted CD19 B cells, clonal B cells from the peripheral blood to the bone marrow. And these are some data we're going to show at the poster at the poster session today. So hopefully, when we finish all of this study, this will help patients to actually be selected for alternative treatment strategies rather than ibrutinib, and then help them not progress to the progression of the disease.